The first part of protein synthesis is called transcription. During transcription, a gene is copied from DNA into a molecule of messenger RNA or mRNA. This process starts with the double-stranded DNA. The DNA is made of nucleotides which contain either guanine, cytosine, adenine, or thymine. These nucleotides are held together with hydrogen bonds, shown here, which always pair guanine to cytosine and adenine to thymine. RNA polymerase, this big enzyme here, is the enzyme that copies the DNA into mRNA. This enzyme breaks the hydrogen bonds in the double-stranded DNA to create two single strands of DNA. Complementary RNA nucleotides are then added to the template strand, here, making mRNA that is almost identical to the coding strand, here. The major difference is that RNA does not contain thymine. Instead, adenine in DNA pairs with uracil in RNA. After RNA polymerase has copied a part of the gene, the two strands of DNA join back together. RNA polymerase moves along the gene until the completed strand of mRNA is made. The DNA remains in the nucleus, while the mRNA can exit into the cytoplasm. Once in the cytoplasm, ribosomes can bind to the mRNA. The ribosome will move along the mRNA until it recognizes a start codon. A, U, G. This signals for the start of translation, where the information from mRNA is decoded to make a polypeptide, or a strand of amino acids that will eventually fold into the three-dimensional protein molecule. Every 3 mRNA nucleotides is called a codon. Each codon corresponds to a specific amino acid. In order to ensure that the right amino acid is brought, there are tRNA molecules with complementary anticodons to each mRNA codon. For example, here we have AUA on the mRNA. That's going to code for isoleucine, or ILE, the amino acid. To ensure that the correct amino acid is brought to the ribosome, there is a tRNA that corresponds to each of the codons. So here we have AUA as the codon, and the tRNA is going to have a complementary anticodon that can bind to it. In this case, UAU. So anytime AUA on the mRNA is there, then isoleucine will be the amino acid that is brought to the growing polypeptide strand. The amino acids get attached together using a dehydration reaction. This will take the OH from the carboxyl group of one amino acid and the H from the amino group of the next amino acid and combine these together to form water. The remainder of the amino acids are then going to form a covalent bond together. New amino acids are brought into the ribosome and attached until a stop codon on the mRNA, in this case UAG, signals for the process to end. This allows the mRNA, ribosome, and polypeptide to separate. The polypeptide will continue on to be folded into a three-dimensional protein that can have one of many functions.